And we seem to have started. So I'll go ahead and cut over to the presentation. And here we go. So when are we finally going to get to the invertebrate animals? Like this fairly recently discovered deep sea swimming segmented worm with tentacles on its head that affectionately got dubbed the squid worm when it was discovered maybe 15 years ago. What do we do with them? You have to ask what defines a true animal, and animals are multicellular, or at least they're descended from multicellular ancestors, but that's not all there is to it, because there's lots of multicellular protists as well. At least one of you, I think it might have been Taylor, already sent me a picture with some green algae growing in it. And if you look at those green filaments carefully, they're multicellular. They're made up of kind of rectangle-shaped cells that just stick together in long filaments, a little bit like the cars on a train. And there's lots of protists that are multicellular. There's even some bacteria that can be multicellular. But what makes animal multicellularity what it is that animal cells are locked together by binding to a meshwork of mostly proteins outside of the cell called the extracellular matrix, or ECM for short. The big component of the extracellular matrix is collagen. You may know collagen because, for example, it's what your tendons are mostly made of. So the Achilles tendon you can feel at the back of your heel is largely made of collagen. There's other proteins like fibronectin and laminin. Elastin is another one. You have lots of elastin in the extracellular matrix under the skin of your face, which gives it its, springy and smooth nature, until when you get old, you lose the ability to make the stuff quite as well and you start sagging. That's all extracellular matrix. Cells stick to extracellular matrix molecules with substrate adhesion molecules called SAMs, SAM for substrate adhesion molecule, and they stick to each other with cell adhesion molecules, or CAMs. The major class of cell adhesion molecules are called cadherins. You also have molecules called integrins. So cells are not just piled up together, they're locked together with CAMs and SAMs and the extracellular matrix. And cells are also linked to each other through specialized proteins that make little pores in between cells called gap junctions. The other thing that defines a true animal is they develop from embryos. And there's a set of genes that's active in all animals at a very early stage of development that's responsible for laying down the basic symmetry of the body. Which end is the head, which end is the butt, which end is dorsal, the back, which end is ventral, the belly, where along the body axis are you going to put whatever organs or structures you're laying down. The genes that do that are called homeotic genes, and there's several different groups of homeotic genes. One of the best known is known as the HOM-HOX cluster. Now, I used to think that HOM-HOX was what you put in your red beans and rice to make it taste really nice and smoky, but evidently I was wrong. The HOM-HOX cluster is a set of genes that is, this cluster is responsible for segment and part identity as you go down the axis from head to tail. In insects, for example, they govern things like where do you put the mouth parts, where do you put the wings, where do you put the legs, where do you put all these structures that have to be put in at particular locations along that axis from head to tail. So just for some visual, this happens to be just some very loose connective tissue. This is the kind of thing you might have under the skin of your own face. You can see the nuclei of the cells, and in and among the cells, you see these fibers of collagen and elastin. So this is just a picture that shows the extracellular matrix really well. In these epithelial cells that have been imaged with fluorescence using a confocal microscope, not unlike the one that we've got at UCA, which maybe you've used or maybe you'll get to learn to use once the damdemic is over. You can see the nuclei of these epithelial cells stained blue, and they're sticking to each other edge to edge like tiles. And what's sticking them together is the specialized cadherin proteins that appear bright red. So you've got low levels of cadherin proteins everywhere, but very concentrated cadherin proteins where the cells are actually sticking together. Integrin proteins look like this. That's a model of them. They bind to the extracellular matrix, and if you look at the one on the right, the one that's unfolded, there's a subunit that's blue and a subunit that's green. And right where the blue meets the green, you can see that little red dashed line. That's where a molecule of the extracellular matrix would fit. So this is kind of the hook that binds the cell to the extracellular matrix. At the same time, they've got a transmembrane domain that extends into the cell and binds to the cell's cytoskeleton. So not only do they physically link cells to the extracellular matrix, but they can activate signaling pathways within the cell. And so cells can sense what kind of matrix they're attached to, and they can respond accordingly. So for example, when in embryonic development, there are times when populations of cells migrate through the body, and they're able to go where they're supposed to because they've got integrins that can sense what kind of extracellular matrix they're dealing with. In fact, this is an example. This is a yawn. Sorry, that's a vertebrate. Sorry, they're very boring, really. But this is a decent example of one. That's probably I'm not sure if that's a fish or not. I don't know what species that is. But what happens in the development of vertebrates and other chordates is that part of the dorsal skin forms a pair of folds which meet in the midline and kind of pinch off a tube. And that becomes the central nervous system, the brain and the spinal cord. Your brain and your spinal cord are basically tubes with very thick, funny-looking walls. As that's happening, as those folds of embryonic skin are pinching off to make the tube, a population of cells buds away from the zone where that's happening, and they're called neural crest cells. And in this embryo, the neural crest cells have been stained green, and neural crest cells migrate away from that newly formed nerve cord, and some of them end up forming ganglia, right? These nerve centers that you've got in various parts of your body. Some of them go on to be involved in the development of facial structures. If you've got a problem with your neural crest cells, you may end up with various facial malformations and deformities, things like that. They can go on to do things like forming your organs of hearing, for example. Those are derived from neural crest, and if you've got a neural crest problem, you can be born deaf. So these wandering populations of cells do a lot of important things, and they know where to wander because they have integrins that bind to an extracellular matrix protein called fibronectin, and they can use that to migrate. So again, this is a very animal thing. You don't have any other organisms that do this sort of thing. that go through embryonic development that's directed in part by the extracellular matrix and the adhesion molecules that enable cells to sense and respond to it. Okay, and then gap junctions are these protein channels that allow ions and small molecules to pass from cell to cell. They can open or close. They're also critical in development, and they allow cells to be electrically coupled. So, for example, cardiac muscle is a big one. The electrical impulse that gets your heart beating actually travels through these bundles of muscle cells from the main pacemaker part of your heart, the sinoatrial node, down some modified muscle cells called, I believe they're called Purkinje fibers, and ultimately stimulate your atria and then ventricles to beat in the correct sequence. And that's possible because your cardiac muscle cells can pass electrical waves because they're coupled by these gap junctions that allow ions to pass from cell to cell. Neat trick. And then finally, homeotic genes are the ones that lay down animal body plans. Up there at the top, these are all fruit fly embryos. So these are all maybe a millimeter long. Up at the top, you've got an early stage of fruit fly development, and each one of those colored bands is a part of the embryo where a particular gene is active. So you've got a green area where one gene is active, a red area where a second one is active, and then the yellow zone where both of them are active. And so you have different genes that are active in different areas. And because genes switch on other genes, they start dividing up the embryo into more and more complex domains. So in B and C, again, those bright areas are zones of an embryo where genes are activated and they're setting up the segment pattern. And then down in D, the colored zones of orange, brown, and black are where a gene in what's called the Hox cluster is active. And the Hox cluster, same thing as the Ham-Hox cluster, that's a set of genes that in the fruit fly governs segment identity. Which of these segments are going to be thorax, which are going to be abdomen, which are going to have legs, which are going to have mouth parts, etc. So you certainly don't need to know all of that, but one of the basic features of being an animal is having homeotic genes like this, in particular, at least a few Hox genes or Ham-Hox genes. It's pretty much the same thing. Animals belong in the last major eukaryote group called the Opistheconta. Conto means, in this case, it means a flagellum. And opisthe is behind. So this is posterior flagellum or flagellum behind. During at least one stage in their life cycle, they've got a flagellum trailing out behind them. Sperm cells would be a good example, or at least many animals have sperm cells that look like this. As we'll see, some don't. Excuse me, gotta sneeze. Be it said that many opistheconts have actually ditched the flagellum completely, and that's true of almost all of the fungi, for example. Most fungi never have a flagellated stage at all, although there are some that do. But if there's a flagellum present, there'll be at least one stage where there's just a single flagellum trailing along behind. And the Animalia are a major subgroup of the opistheconts. So are some protists, like this weird little amoeba called Nuclearia, which looks vaguely like a fried egg. Despite its amoeba shape, the DNA says that it's not closely related to the amoebozoa. It doesn't go in with, that first group that we talked about. it's actually more closely related to fungi than to anything else. So that's an episthecont by common ancestry, even though it looks like it should go off with the amoebas. It's not closely related to them. There's another funny little amoeba called Capsospora of Charzaki. So that's Ovchashaki, Polish name, Ovchashaki. 5 microns wide and a symbiont in a freshwater snail, because somebody has to be. Might not be especially interesting, except it turns out it's a very close relative of the animal lineage and has some features in common with animals, especially integrin proteins. What they're doing there, I don't know. There's not an awful lot of people that study Capsospora of Chashki, of Chashaki. Not a lot of people can pronounce Capsospora of Chashaki anyway. But nonetheless, it's one of these epistheconts that isn't in the Animalia, but seems to be closer to the Animalia than to any other group. And then, of course, we've got fungi, which we're not really going to cover in this course. So yeah, here we've got some fly agaric mushrooms. Not that you really wanted to know this, but part of my role as a professor is periodically grossing students out with weird stories. This one is actually hallucinogenic. I don't think it grows here. It's native to Northern Europe. And you have to prepare it right. You know, if you eat it, you'll see God. Eat too much of it, and you might be seeing God permanently. But if you prepare it right, it's comparatively safe, although I'm certainly not telling anybody to experiment. But the hallucinogenic compound in it, called muscimol, is not metabolized very well by the body, and it's excreted by the kidneys, which means that in some tribes in Siberia, the local shaman, or the local spiritual practitioner, the guy who goes off and talks to the spirits, will eat some of these mushrooms and hallucinate. And while he's hallucinating, somebody follows him around with a bowl, and they collect his urine, and then somebody can drink that and go on very much the same trip. And you can apparently have the same batch of mushrooms passing through multiple excretory systems before it finally wears out, because the body doesn't break down muscimol very fast. So yeah, you can actually filter this through several people before it starts losing effectiveness. So that's your horrifying fact to freak out your friends and relatives at the dinner table or on Facebook or whatever you're using for the day. Yeah, that's your daily freak out. Right. There's one little group called the Microsporidia that they used to consider to be parasitic protists. Once it got possible to sequence DNA, it turned out that what they actually are is very stripped down fungi. And I mention them because there's a chance you might see some of these in your insects. They can cause disease in a number of insects. This particular one was actually extracted from fire ants, and people are interested in it because people are interested in anything that'll kill fire ants. Trust I don't have to explain why. There are some that can cause disease of honeybees and bumblebees, and that's economically important. There's some that are looked at as biological control agents because they'll kill mosquito larvae. They can parasitize other critters as well, including one species called Enterocytozoon bunusi that doesn't normally do very much in people, but it can cause pretty severe wasting and diarrhea in immunocompromised patients. It was one of the rare infections that turned out to be surprisingly common in people with full-blown AIDS. which mercifully doesn't happen so much now that we've got medications that can manage HIV infection. But people with HIV used to get all sorts of infections that uninfected people never got, and this was one of them. They're also neat because they've got the smallest genome of any eukaryote. You've got about 3.2 billion nucleotides in your genome. One called encephalidozoon makes do with 2.3 million. So these are eukaryotes, but they have stripped down to pretty much the absolute minimum we think that you can have and still be a eukaryote. And again, I mention them because there's a chance you'll see them. They're not real big. They're going to be about 5 microns and clustered like this, and probably hard to tell from other little tiny spheres that you might see. But if you spot them, make a note of it. This is interesting. But yeah, I mention them because there's a chance you'll see them. But what they actually are is very, very weird fungi. So what about the Animalia? The closest living protist relatives to Animalia belong to a group called the Choanoflagellata. Choanon is apparently Greek for a collar. So the Choanoflagellata are the collar cells. They are animal-like in many ways, and some of this I'll show you in a bit. They're animal-like because they contain cadherins, the cell-to-cell adhesion molecules, and they also have these surface receptors that are critical in cell-to-cell signaling that animals have and that choanoflagellates also have. These receptors are called receptor tyrosine kinases. So they've got cell adhesion and cell signaling molecules that look like animals. And of course, they've got very similar gene sequences to animals. They're not uncommon in freshwater, and there's a chance you could spot some in your pond water. The problem is they're really small, and they're not that easy to observe, especially if your highest power is only 400x. They're basically just kind of rounded to oval cells. There's a single long flagellum, but surrounding the base of that flagellum, there's a ring of little projections of the cell called microvilli. Remember, a villus is a little projection of something. Your intestine has lots of villi increasing its surface area. Microvilli are, really small villi, and you'll have a ring of microvilli coming out of the cell, encircling the base of the flagellum like a collar. And when the flagellum beats, it may enable the cell to swim, although some choanoflagellates are actually sessile. They don't swim. Regardless, the flagellum beating pulls water currents through that collar of microvilli, and they sieve out food particles. That's how they feed. So there's a diagram of one. The cell body is kind of nondescript and, round to oval. Projecting out of it, you have that ring of microvilli, and then the flagellum comes out in the middle of the ring, and as it beats, water is pulled down, through the microvilli and then upwards. And the microvilli can sieve out tiny food particles that way. And that's what one of these looks like under the light microscope. This one, Acanthocorbus, is only about 6 microns long, meaning under your scopes at 400x, it's still pretty much going to look like a dot. Maybe a big dot, not as small a dot as a bacterium would, but you're not going to see much detail. And even here, that's a picture that's been taken using a special microscope technique called interference contrast. The body of the cell itself is the thing that looks kind of like a carrot. And around the base of the carrot, if you like, you can see that circular thing. That's the collar. And the microvilli are so close together that you can't tell that collar is made up of microvilli. It just looks kind of like a little solid cone-shaped to circle-shaped thing sticking out of the cell. And then, of course, from right in the middle of it, you can see the flagellum projecting over to the right. There's another one. Monosiga is stalked right there. That's the one on the left. And the cell body is kind of oval. There's a long stalk growing out of one end. And at the other end, you can see those two things sticking out. That's the sides of the collar. And you can just barely make out a little bit of the central flagellum inside the collar. If you compare that to a related genus shown with SEM called Salpingoica, you can see the stalk, the body, and in that one, you can see that that collar is made up of microvilli with a single flagellum sticking out. Choanoflagellates can actually be pretty common in the oceans. The ones that I've heard of tend to be found in polar waters. I'm sure there are some in tropical waters as well, but these are two Antarctic species. And in these Antarctic marine species, the cells actually secrete these little silica bars and can actually stick them together to make a basket-like structure called a lorica. Lorica, by the way, is a word you're going to see again. I might not even present it here, but it does pop up when we're describing other critters. Lorica is the Latin for breastplate armor. If you've ever seen movies about Roman soldiers where the centurion is wearing this, brightly gleaming bronze chest protector that makes him look like he's got massive pecs and a six-pack, that's a lorica, the breastplate. A shirt made out of chainmail could also be called a lorica. So a lorica is just a term for chest armor, and it's also used in invertebrate zoology to mean any animal structure that reminded somebody of armor. So the lorica right there is this silica bar structure. Right. Where things get weird is when you discover that some choanoflagellates are single cell. They'll spend all of their lives at just one cell. But there's others that form colonies. This is one, Spheroica. That's a whole bunch of little collar cells all packed together into a hollow sphere. Interesting. And there's a very rare genus, Proterospungia, that's not been described very well because it's not often seen, but it's got flagellated cells on the outside. On the inside, it's got amoeba-like cells, and all of these are in an extracellular matrix. So this is cool. We've got an ECM, and we've got cell differentiation, different types of cells for different functions. Proterospungia's got two different cell types. You've got roughly 200, although it depends on who's doing the counting. But one of the things that makes animals animals is not just multicellularity, but differentiation of the cells. It's not an absolute cutoff because you get this as well in seaweeds and things like that. But you're seeing it here as well. And Proterospungia may give us some insight as to what the ancestor of all animals might have looked like. It wouldn't be identical. Proterospungia is not ancestral to anything because it's 750 million years too late, but it might give us some insight into what a common ancestor of animals might have looked like and might have been able to do. And the reason why that matters, here's another colonial choanoflagellate. There you can see the collar much more clearly. You can even see separate microvilli in the collar. There's pretty much the same species seen here using scanning electron microscopy. You can see the collar of the villi really well here. And they're dead ringers for a cell type found in the simplest true animals, the sponges. What you're looking at here is specialized cells inside a sponge. And what you ought to be able to recognize really easily now is you can't really see most of the cell body. It's kind of buried. But from each one of these cells, you have a collar of microvilli. And growing out of the center of that collar of microvilli, you've got a flagellum. It's exactly like choanoflagellates, but it's a cell type that's found inside the simplest of true animals, probably the phylum Porifera, otherwise known as the sponges, like this giant Zestospongia Caribbean vase sponge that you see right here. Okay, I've got 1038, but that was kind of a big... Hi, sweetie. My wife is making funny faces at me.
You taking a break?
Just about to. But that's a pretty good place to pause, and then we can go back and look at the sponges again in a bit. Let me know if I'm going too fast or too slow or too boring, or if I need to do magic tricks or something like that, or cause explosions. I'm having a hard time getting feedback that I'd usually get from tone of voice and eye contact and things like that. So if
I think we're missing the explosions.
Oh, you want explosions?
Oh yeah, we're missing explosions. Maybe bring Jack the Rabbit in there too. Who knows?
Bring who?
The rabbit thing, you know, the cap and the rabbit. I don't know what it's called. I don't know what they call it.
The cat and the rabbit?
The cap and the rabbit, like the magician thing.
Oh, right. Okay. I'll work on that. I'll see what I can do.
Sounds good.
Yeah, problem is every time I pull a rabbit out of my hat, the cats kill it and eat it. So, but all right, I'll see what I can manage. But seriously, since I can't get feedback from watching your facial expressions and your eye contact and things like that, make sure if something isn't clear or I need to go over it again, or you really wish I'd shut up and move on to the next topic, make that known to me. I'll be as interactive as I can, even if it's not really an adequate substitute for doing this in person. So I'll go ahead and hit stop recording, and then you can cuss me out if you need to.